My lab is really interested in network theory in all its possible applications. So we have worked on networks to describe patient similarities. So for example, if we have molecular data sets coming from patients with different diseases, we have recently, well, not so recently, but not so far away, we published a paper looking at comorbidities between different diseases and how we can sort of predict them or at least interpret them based on the transcriptomic similarities of profiles of the alterations that are created by the diseases in the transcriptomic profile of the patients. So that was a recent study more on the network medicine, let's say, approach. And then we use networks to also represent tissues and actually cells in tissues. So we have another, this time quite recent paper. We have developed a software called Tisserand with my postdoc Alexis Coulomb. And basically, this software allows us to turn a biological image, it can be an anatomopathology image or any microscopy image actually, of cells, into a network where each cell is a node in the network, and we can represent their location in space and their interrelations due to their contacts and proximity. So these type of models, we are now applying them to consider spatial transcriptomics data sets or spatial proteomics. It's a really general framework that can be applied wherever you have spatially defined phenotypes, either from an image or from any other data set. So that's one application, and actually this is what we use to merge data sets that come from different platforms. So once we have the phenotypes associated to the cells, we then also represent the spatial distribution of these cells in the tumor microenvironment. And this helps us understand also the interactions between the cells. So in the future, we hope that this will feed into these in silico models so that we will be able to really specify the statistics of the world in which these agents live, which is basically the characteristics of the simulations. We also made some applications of network theory in epigenomics. So I'm also very interested in chromatin structure, and we have recently continued our efforts to understand and integrate different epigenomic processes and data sets on networks that represent the chromatin structure inside cells. And there might be some application in cancer, but it's a little bit too basic for the moment. But yeah, we love network science.